Okay, so welcome everybody to the extra session. And if you have been following the sessions that I was conducting every Monday, actually, during the course of this, lecture, then you know that the part that was left for us to discuss was the sensory systems, right? So this is anyway something that I have to discuss along with the auditory and visual senses that have already been talked about in the course. I have tried to include a more general kind of introduction for all sense organs and talk a bit about all of these as well, right? The five main senses and the sixth, that is a proprioception, that is the from within the body when you get senses about what is going within the body, which is proprioception, right? So these are things that I am anyway going to discuss today. Apart from that, I wanted to ask you guys, like I hope that your assignments and everything have been going smoothly for this course, and it would have come to an end already, right? Your 4-week course would have been over by now. All the course material would have been released. I think you might have also crossed the deadlines for your assignments. If there is any, problem or confusion with the concepts, then also you can let me know. I'll try to take that up also. I'll try to clear it out. Uh, and if it is not possible right now for me to clear it up, then maybe I can respond over mail also to you guys, right? So, yeah. As of now, what we'll do is we'll begin with the discussion of the sensory systems, right? If you guys have any confusion or problem, you are free to speak up, okay? Right. So, yeah. I'll just share my screen once more. Yes. Okay. Um, so one last time, I'm Arveen, and I have been your TA for this course Neurobiology. And this particular session is an extra discussion session in which we are going to take up the sensory systems, right? So what we'll be dealing with is we'll first understand what are the different kinds of senses, right? So one of the senses, for instance, is vision, right? So this is one of the, uh, what representations that have been taken from your book. And over here, you can see that one of them is what the sense is, and the other, representation just by its side or on the right is basically what is aiding us to sense that particular kind of thing. Right. So, for instance, first is vision, right? Vision or sight, right? How you see things. Now, that has there are some neural cells, rods, cones, and multiple others also that are involved in your vision or visual perception or vision sense, right? Perception is a much bigger, broader topic. So vision sense is what is controlled by your cells in the retina, right? Two of those cells that you commonly have studied are rods and cones, right? Then comes smell, for instance, right? So there are multiple cells that are again, neural cells that are lining the nasal epithelium. And this olfactory, olfactory yeah, nasal epithelium, olfactory epithelium, that is between your nose and your brain, right? And that is what is being responsible for you to be able to sense smell, Similarly, there are multiple taste buds in your tongue that are helping you sense different kinds of tastes, right? So there will be some kinds of taste buds that will be responsible for the sour kind of taste. Some others will be responsible for the sweet kind of taste that you get, right? And multiple others, right? Depending on what kind of taste you get, and a combination of these get activated so that you can differentiate between the kind of taste that you are feeling. Then comes touch. There are multiple pressure sensors, receptors, mechanoreceptors that are present on our skin, especially of our fingers, because they are the fingers are the most used for perception, for sensing, right, through touch. And so these kinds of receptors, they are very densely present in our fingers, right, of our hand. Then comes thermal senses again. There could be some kind of pain receptors, some kind of mechanoreceptors that can help us sense different kinds of thermal things, right? Whether it is hot, cold, etc. Then again, pain sensing is one of them. So pain sensing again could be due to some kinds of receptors or some kinds of neurons that are present. And it is also said that taste, in case of taste, the spicy sense is also a pain, receptor that is getting activated, right? So there are multiple other places also where these pain receptors are, right? It could be in case of thermal senses also. If we feel very cold or very hot, it might be because our, we are seeing that we are touching something, icy or some flame, but the what our body is feeling is pain, right? So over here also there's some kinds of pain receptors might be present. Then comes hearing, which again is another, major kind of sense. Yeah, it is another major kind of sense through which you again sense what is your surrounding, what is the kind of sounds that are coming from your surrounding. It is very, very important for communication also, right? Then comes balance, right? And proprioception. Now, both these are a function of balance. How it occurs? It occurs due to this particular, kind of follicles that are present within these three semicircular canals in the inner ear, right? So that is balancing our body. That is what is maintaining us erect in space, right? Then there is proprioception, of course, which again, as I told you, is your knowledge of where your own body parts are, or what is the kind of changes that your body is feeling, right? Internal changes, not the external ones, right? So that is what is proprioception. So a little bit about each of these we are going to discuss about. First, over here you saw there are different kinds of modalities that are linked to different kinds of senses, right? Over here, let us classify these modalities or sensory receptors, right? So there could be three major kinds of sensory receptors. There could be chemoreceptors, photoreceptors, or mechanoreceptors, right? So these are the three major types, right? What are the chemoreceptors? For instance, there is some chemical, there is some odorant that is coming and sitting on the receptor, or it is, inducing some chemical change in the receptor, due to which either the morphology is changing or some kind of a signal transduction is getting initiated, and, whatever effect has to happen is happening, right? What is a photoreceptor? Over here, a single photon of light is what is activating these receptors, or what is setting into motion whatever, signal needs to be transmitted. So that is photoreceptor. And then there is mechanoreceptor. Now this is being, activated or stimulated by the pressure, right? So this is a kind of receptor that is sensing pressure. And if, for instance, pressure is getting applied, it is changing conformation, it is getting opened up, and again, the signal is being transmitted, right? So for instance, uh, before we move forward, we will also study a few terms that we will eventually need, right? One of these terms is receptive field. Now, what is receptive field? A receptive field of a sensory neuron is basically the spatial domain, like in space around it, uh, in the sense organ where stimulation excites or inhibits the neuron, right? So, for instance, there is one neuron. If we stimulate this neuron, it will basically transmit the information to our brain that some kind of pressure is being applied on me, right? Or on this region of the hand, right? So what is the receptor field? Receptor field is the space in which that neuron can be activated, right? It is possible that, for instance, how do I say? So, for instance, this is the neuron, right? This entire thing could be, or this entire thing could be an area. Okay, this is more important. This is easier to explain. So, this entire thing could be an area and this place wherein something is written, right? That could be the receptive field of a neuron, right? So if I apply pressure here, this we are considering that a single neuron is innervating this space, right? So if I'm applying pressure over here, nothing will happen. No, signal will be transmitted to my brain. If I'm applying pressure over here again, no signal is getting transmitted. If I'm applying pressure over here again, no signal will get transmitted. But if I apply, pressure here or here or here, all this is within the receptive field of the neuron that I'm talking about, right? So whatever pressure I am applying, that signal will get transmitted to the neuron. So that is basically what is the spatial domain, and it is known as the receptive field, right? Then if we classify these sensory receptors, we look at it one by one, right? Okay, I'm unable to zoom it. Yeah. So one by one, we'll classify the sensory receptors, right? So if we are saying the visual kind of sensory system is what we are talking about, the modality that it is facing is vision, right? So what are we seeing? Vision, right? What is the stimulus over here? It is photons or light, right? So what will be the kind of receptors that will be present in this kind of system? It will be the photoreceptors because light is what is activating or stimulating them, right? And the cells that are responsible for this photoreception or the photoreceptors themselves are the rods and cones that are present in the retinal layer, right? Then comes auditory senses, right? So the auditory sensory system has one function which is to hear, right? So hearing is the modality. The stimulus is sound. Now, what is sound? It is simply pressure waves, right? And the class of receptors that are responsible for this kind of sounds or perceiving these pressure waves are mechanoreceptors, right? So some kind of wave is getting generated and it is being received by these receptors. So it is causing a mechanical pressure, right, on the receptors. And what are these receptor cells particularly? They are the hair cells in the cochlea, right? Again, cochlea will be in the inner ear. Then the other sensory system is vestibular, right? Now, what does this mean? It means, the perception of head motion, right? So that is the modality. What will be the stimulus for this? Gravity can be the stimulus for it, right? So gravity is basically pulling us towards the earth, right? But our head is still remaining like this. We are not bent like this, right? There should be some reason for that, right? Then acceleration, for instance, if you are sitting on a scooter or a bike, especially if you are on the back seat, right? If you are not riding it, if you are on the back seat, if somebody accelerates quickly, you are taken aback like that, right? So why? Because when you are riding it, you are more prepared that this is when this thing is going to happen, and you know how much you are accelerating. You don't get that much of a shock. But if you are sitting at the back, you are not prepared because you are not making the call, or your brain is not making the call of how fast the acceleration is going to go, right? So all of a sudden you are taken aback, right? So acceleration and head motion, these are also stimuli for this kind of vestibular perception, vestibular sensation, right? What are the receptor classes? Again, the receptor class is going to be mechanoreceptors, and the receptor cells will be the hair cells in the vestibular labyrinth. Now, this is also inside the inner ear as well, right? And the cranial and dorsal root ganglion cells with receptors in skin. Oh, that is for somatosensory. It is not for this. So for vestibular, it is in the labyrinth itself. Right? So, yeah, then there are multiple kinds of somatosensory sensations, right? Now, this could be touch, it could be proprioception, it could be pain, itch, visceral, etc. Right? So, for instance, touch, when we are talking about it, what is the kind of stimulus that we are getting? It is skin deformation and motion, right? So, if I'm moving my hand like this, it is motion. If so, my finger is basically coming in contact with the air around it, and some kind of a pressure is getting applied. It is very very minute, but I know that my hand is moving in a certain way, right? Then it could be skin deformation. For instance, even if I'm touching my two fingers together, the skin is coming in contact with one another, right? So some kind of a minute level, cellular level deformation is happening. And what is the kind of receptor that is sensing it? It is again a mechanoreceptor, and it is present in the skin, of course, right? Then comes proprioception, right? So this is what we have talked about earlier. The kind of stimuli are muscle length, muscle force, and joint angle. And again, mechanoreceptor is the kind of receptor, and it is present in muscle spindles and joint capsules, right? Then comes pain, right? So pain could have a lot of stimuli, any stimuli that is harmful for us generally. It could be thermal, mechanical, or chemical, right? So some chemicals burn you, some temperatures they can burn you, some mechanical stresses can basically scrap you, scratch you, something like that, right? So multiple different kinds of stimuli could lead to pain, and there are multiple different kinds of receptors that are responsible for pain, right? So all the three receptor classes that we talked about have some role or the other in pain sensation, right? It could be context specific, right? If it is due to some burning flame, it will probably be a thermoreceptor and not a chemoreceptor, right? So it will be context dependent, and it is expressed in all tissues except in the central nervous system, right? So this is another very fascinating fact that you might have come across already, that for instance in a brain surgery, right, once the cranial window is created, that is once the skull has been penetrated, right, the person is generally awake for the surgery. And even if the surgeon is basically poking around in different regions of the brain or doing whatever, the person does not feel any kind of pain because there are no pain receptors in the brain. Right. So again, it is pain receptors are not present in the central nervous system. Then comes itch, right? What is the cause of this itch? It is histamine, right? That is why for itching, for these kinds of insect bite related inflammation or allergies, we take antihistamines, right? Because they help cure this particular or subside this particular stimuli, that is histamine. And the kind of receptor over here is, of course, a chemoreceptor because a chemical is involved, right? And it is more commonly expressed in the skin, right? Because that is where we feel the itch, right? We don't feel an itch inside our heart or itch inside our muscle. That is not what happens, right? Then visceral, right? Apart from the pain receptors, the visceral kind of sensation, which could be either thermal or mechanical or chemical, right? Again, there could be all three classes of receptors that are involved in this, and the kinds of places it is expressed in is gastrointestinal tract, urinary bladder, lungs, etc. right? Then there are two other modalities that are there. One is the gustation, two other senses that are there. One is the gustatory sense, which has the modality that is taste. It is, transmitted through chemicals, and which the kind of receptors that are involved over here are chemoreceptors, which are present in the taste bud. And the final kind is the olfactory, the modality for which is smell. It is transmitted through odorants, again, which are kinds of chemicals. So the receptors over here are chemoreceptors, and they are basically present in the olfactory sensory neurons, right? So these were the different kinds of sensory receptors, their modalities, what are the receptor classes that are present, and what are the stimuli and receptor cells, right? And then we will come on to discussion about each individual kind of the same sensation, right? So first we will begin our discussion with vision, right? So it is one of the most time-taking ones to talk about, and it is very, very elaborate, right? So let's see how we'll understand this. So when we talk about vision, uh, let's read this, right? Although the lines, eyes play an undeniable crucial role in vision, right? The brain is also involved in actively constructing a visual representation of the world that is not a literal reproduction of pattern of light falling on the eyes, right? For instance, a simple example would be if you see a cloud in the sky which is shaped like a star, right? Like it is closely resembling a star, like that kind of that pentaploid kind of star that we have generally drawn when we were little, right? Or the star that goes on top of the Christmas tree, that kind of a star. So what you are actually seeing is a cloud, and that cloud is in a it is just a cloud, right? Now, what shape it is in, you have already registered in your mind, or you already know in your mind that, okay, this kind of a shape is a shape of a star. So you are now linking it to that. So you are now saying that that is a cloud that looks like a star, right? So over here, what your eye is seeing is simply a white, blob on a, blue background, maybe, right? But what your brain is perceiving is that that is a cloud, and that cloud looks like a star, right? So that is something different. So it is not just something that we are seeing, but it is also, our brain is also actively involved in perceiving what we are seeing, right? So here are a few examples of that we'll discuss very soon, right? So for instance, when we talk about human perception of colors, it results from simultaneous activation of three different classes of photoreceptors in the retina, right? So most of the colors, when will you see it? During the daytime, right? During night time or during dark, you will not be able to distinguish colors that well, right? They will be different shades of gray, or different shades of, dark, white, gray, black, something like that, right? But during the daytime is actually when you will be able to differentiate between different colors, right? So during the daytime, the three, types of photoreceptors that are actually active are the cones, right? So they are the blue cone, the green cone, and the red cone, right? So there are only these blue, green, and red cones that are active, but depending upon what is the combination of them that gets activated, how long one of them is activated, or how many of them are getting activated together, finally decides what is the color that we are seeing, right? And during the night time, which is a photoreceptor that is activated, it is a rod, right? Which has a much higher range of color perception, right? So for instance, if these many green, not so many blue, and these many red cones were activated, then we would see the color yellow, right? So it is kind of a pyramid that represents that. Okay. Then again, it is not just about color when we are perceiving something, right? There are spatial properties of scenes and objects which are conveyed by population of neurons, each of which represents small component of the image, right? So what it means is that, for instance, we are trying to see something, right? If multiple neurons are located in that region where the light is falling, we will be able to see a clearer image. But if lesser neurons are actually present in that region, we will be not be able to see those many images, that sharp an image. Okay. So, for instance, you can relate it with the number of pixels in the image, right? So this is the same image that has been recreated with different number of pixels, right? So in the first one, there are 20 by 20 pixels, right? So roughly around 400 pixels, right? So when we see this image in a 400 pixel, it looks like a blur to us, right? Then there is this one in which there's a 60 by 60 pixel size, right? So it will be some how many 3600 pixels, right? So over here you can see it is clearer than the previous one, but not as clear as one might like, right? Then over here we have 400 by 400 pixels, right? Now that is going to be a lot. It will be some 1,60,000 maybe, right? 16 or 16,000. Yeah, 1,60,000, right? So those many pixels. So you are seeing that from around 400 to around 3,600 pixels, how clearer the picture has begun and become and so on, right? So that is what is happening with our I also, right? For instance, you have studied this as a part of physics also, and maybe as a part of biology also, but mostly physics during your, 10th and 12th, that when the light is falling, right, when the lens is proper, for your eye, when the lens is proper, the light falls on a certain region, right? Now, this region has high density of neurons, which is why you see a clear image like this one, right? But if there is some kind of, mishap in the lens, or such that the light is falling before or after where it is supposed to fall, you will not see a clearer picture, because maybe the density of neurons in the place where the light is falling is not as bright, right? It is not as much, right? So for that we use external lenses, which are our glasses, basically our spectacles, which can correct that angle so that the light again is falling on the region where there is a high density of neurons that is present, so that we can see a clearer image, right? So I think most of you might have already studied the physics behind it, but this is the biology behind it, right? So that is why, with the help of the spectacles, we can actually see a better picture, right? Then we come to another discussion, which is of a discussion regarding sensation versus perception, right? So this is what we were talking about when we took the example of the cloud as well, right? So sensation is something different. It is simply what we are, sensing. So it could just be some sound that we are sensing, or something that we are seeing, right? But perception means making meaning out of the sound that we are hearing, or making meaning out of what we are seeing, right? So sensation refers to effects of stimulus on the sensory organs, whereas perception involves elaboration and interpretation of the sensory stimulus based on, for example, the knowledge of how objects are structured, right? So over here, what will be sensation and what will be perception, right? So sensation would be that this is one structure, this is one structure, this is one structure. Then there is one bracket over here, one bracket over here, one bracket over here, right? So these are the 6 structures that are present, right? That will be sensation. But first, when we looked at it, what did we see? Did we see that only? So that is a question. Do you automatically perceive a white triangle that isn't really there? Right? We automatically thought this is one triangle, it is lying on top of this other triangle, which is lying on top of these three circles. Right? So that is not what it is, though, right? So this is called the uh, Caniska, Canissa illusion, right? So this is what it is. This is the difference between sensation and perceptions, right? So once when anybody has created this, they have actually kept, six different pieces, but they have arranged these six different pieces so that they look like three circles, one solid line triangle and one solid color triangle, which is lying on top of these circles, so that they are becoming kinds of they look like quarters of a circle, right? Yeah. So that is what is sensation versus perception. Now this is another example, right? So what do you see over here? By default, what we'll see is that there are 1, 2, 3, 4, 5, 6, 7, 8, 8 circles, and on top is placed a cube, right? Or some people might see it as a rhomboid also, right? Again, this is another example. Now, this is something that looks like, scattered pieces of some yellow color, right? But and this is going to remain same in the next image, right? So the pieces over here are still the same. They are still scattered and they are exactly the same as the last image. But now what do we see over here? We see the letter B, right? So that is the difference between sensation and perception. Over here, because it has something on top of it. It is not just this image. This image has something on top of it that looks like spilled ink. We think that the letters underlying it, the things underlying it is a letter which is B, right? So that is what is perception, and that is where our nervous system is playing the most crucial part, right? When we talk about again vision, there are these neural cells that are associated with it. So first we'll understand the structure of the neural cells inside the eye, right? So this is the eye. You know that there is a pupil, there is a cornea, iris, which is the colored portion of your eye, the lens that is present, the ciliary body, and the ciliary muscle that is the keeping your eyes open, not letting it droop, etc., and the conjunctiva or the clear layer, right? Then there is aqueous humor over here, there is vitreous humor over here, right? Apart from that, there are three layers that are present, right? They are the sclera, the red, okay, one, yeah, there is a sclera and retina that I mentioned over here, right? The retina is this innermost layer, and the retina is the layer wherein all these neural cells are located, right? And it is connecting to the optic nerve, which is transmitting whatever signal these neural cells and photoreceptors and are getting to the brain, right? So retina is the internal surface of the eye that contains specialized photoreceptors that convert or transduce light into neural signals, right? So they are, what is the stimulus over here? It is the photons. And these are photoreceptors that are converting these signals from the photons of light to information that can be processed by the brain, right? Then these photoreceptors are made up of two types of cells, the rods and the cones. The rods are specialized for low levels of light intensity, such as those found at night, whereas cones are more active during daytime and is specialized for detecting different wavelengths of light from which the brain can compute color, right? Now, if we look at the retina, right, if we try to separate this layer. Now, the other layer was choroid that is not mentioned over here. There is sclera, choroid, and retina. So you can see retina is the most elaborate layer. It has multiple cells. It has the pigment epithelial cells, the rods, the cones, horizontal cells, bipolar cells, amacrine cells, ganglion cells, etc. Right? So it has multiple cells and multiple layers within it. Right? So there is already a stage of neural computation that takes place in the retina itself, right? So why? Because signal has to transmit from these rods and cones to the optic nerve. In between, there are so many cells that are lying, right? So the signal already goes through some level of processing during crossing these cells. For instance, the bipolar cells over here, as a name suggests, they are bipolar. On both sides, they have, signal transduction capabilities. They are types of neurons that behave in one of the two ways. Either they detect light on the dark backgrounds, which is on, or they detect dark areas on light backgrounds, which is off. Right? Then there is higher level of processing by retinal ganglion cells over here, which have more complex set of on and off properties, right? Most of the retinal ganglion neurons have a particular characteristic response to the light that is termed as center surround receptor field, right? We are going to skip the center surround. I have kept those slides, but we are going to skip that for now, in the interest of time, right? So the term receptive field over here also denotes the region of space that elicits a response from a given neuron, right? On one intriguing feature of the receptive field of these cells and many others in visual system is that they do not respond to light as such. They respond to differences in light across their receptive field, right? Only this part I'll explain, right? So it will not just be the photon coming in that it will respond to, right? It will be the, it, that it is able to distinguish a particular light coming from its background, right? That will be the receptive field, right? For instance, if at night you go to a very dark room and you're not able to see anything, right? That is because your rods or your photoreceptors are not able to distinguish the light between different objects in the room, right? But if you go into a dark room and you can still make out, okay, that is where the window is, that is where the door is, this is where the table is, this is where the bed is, right? Why are you able to make it out? Because there is some kind of a difference of the between the light that is bouncing off from these places and the light that is in the background, right? So you are able to distinguish it from the rest of the darkness in the room, right? So that is what is receptive field. This center on off we are not going to discuss right now, right? Yeah. Then there is also something known as blind spot, right? So what is this blind spot? It is basically a hole in the retina. Why is this present? Because this is where the optic nerve is getting connected to the retina, right? So the retinal layers or retinal cells are not present over here, right? There is kind of a gap. There are no photoreceptors over here because of this, junction that is giving way to the optic nerve. That is why it is known as blind spot, because whatever light will pass on it, it will not be able to perceive because it has no photoreceptors, right? So the output of the retinal ganglion cells is relayed to brain via the optic nerve. That is what this optic nerve will do. The point at which the optic nerve leaves the eye is called the blind spot because there are no rods and cones that are present there, right? No cells at all that are present over there. Now, this particular slide is for, for you guys to experiment uh, about where or how we can test this blind spot, right? So over here you see two objects. One is a circle and one is a plus sign, right? So whenever you will keep a device, so there will be a recording also for this. You can pause the recording in on the slide and you can test it out, right? Whatever device you are holding, if you are trying to focus on any one of this, right? Suppose I am focusing on the plus, right? Keep your eyes focused on that plus and try to, move yourself away from your device slowly or gradually bring the device closer to you. There will be like once you're looking at, one of these, you will still see the other thing in your peripheral vision, right? But there will be one particular, range at which you will stop seeing the peripheral thing. completely, or you'll altogether not see it at all. Now that is because it will, fall on your blind spot, right? So that is just how we understand where the blind spot is, right? But how we test that, oh yes, there is a blind spot, right? Then there is another region which is opposite to that of the blind spot, which is a fovea, right? This fovea is actually the region where there are maximum number of neurons that are present, right? And as we had already studied, if the neurons density is high, then the picture will be sharper, clearer, right? It is like having much more pixels in within an image, right? So the image that we will ultimately obtain will be very, very clear, right? So that is what this fovea will have. It will have the highest concentration of cones that in one point, and the level of detail can be perceived at the greatest at this point, right? So it will be almost equivalent to the third image in that pixel thing that we had discussed earlier, right? Rods are more evenly distributed across the retina, but they are not present at the fovea, right? So this will have more of cones that are present, right? So very sharp images it will create. Then we come towards the perception of this vision, right? So primary visual cortex, which is again present inside the brain, and the geniculo-striate pathway, right? So these are the two things that we are going to now understand what they are, right? So the dominant visual pathway in the human brain travels to the primary visual cortex, right? So the optic nerve is going to the brain, it is going somewhere, right? So it is going to the and supplying information to the primary visual cortex, which is present at the back or the posterior of the brain over here, right? Via a processing station, which is called the lateral geniculate nucleus or LGN. Right. So the optic nerve is traveling to the back of the brain, but it is going via this particular lateral geniculate nucleus. Right. So this is what is happening. Right. This is where the vision is getting sensed. Optic nerve is taking whatever sense it is getting via this optic tract, which is passing the LGN or lateral geniculate nucleus to the primary visual cortex, that is towards the posterior end of the brain, right? And this pathway of the primary visual cortex from the retina to primary visual cortex is basically known as the geniculo-striate pathway, right? So the neural representation in the lateral geniculate nucleus divides up the information on the retinal surface in a number of interesting ways, right? So what will be this information? The objects that are seen from the right side of the space are termed as right visual field. They will they will be carried towards the uh, lateral geniculate nuclei, and they will be perceived by the left primary visual cortex, right? Similarly, this information is segregated into 6 different neuronal layers of the structure, 3 for each of the eye, right? And the layers of lateral geniculate nuclei are not only divided according to the eye, that is left or right, but contain a further subdivision. You can see there are 2 within this also. Now, what are those subdivisions? The upper 4 layers have small bodies that have been termed as parvocellular or P layers, whereas over 2 layers contain larger cell bodies that have been termed as magnocellular or M layers, right? So this is the layers within the retina that we are talking about, right? So the parvocellular or the P layers, they respond to detail and are concerned with color vision. Whereas the magnocellular cells more sensitive to movement and color, and they respond to larger areas of visual field, right? So magnocellular, a good example would be, for instance, a train moving, right? Or a car moving. Parvocellular would be a good example for this kind of a vision, or this kind of a thing getting more stimulated is probably you looking at a painting, right? Or you were trying to figure out the details within this schematic or diagram, right? So more recently, a third type of cell has also been documented. It is a K or conio kind of cells in the lateral geniculate nucleus itself. Uh, which lies between these magno and parvo layers. Right, these cells show much less functional specificity that magno and parvo, and have a different pattern of connectivity also. In 1981, multiple properties of these primary visual cortex area were elucidated by these two scientists, Torsten Wiesel and David Hubble, both of who were, one was a Swedish neuroscientist, the other was a Canadian neurophysiologist, right? And they were awarded Nobel Prize in Medicine in 1981 to, honor what or uh, yeah, to honor their research. Yeah. So they had found that particular neurons in the visual cortexes of cats and monkeys, which were in areas in the brain responsible for processing visual information, didn't respond to simple points of light, but rather to lines, and in particular lines and contours with specific orientation. Right. So if, for instance, uh, somebody was showing only light to it, this, processing area might not have, been active. But if somebody was trying to show anything resembling even a little bit of a pattern, these areas started functioning, right? So some neurons responded to horizontal lines, while others responded to vertical, and still others to orientations in between, right? And there were different kinds of neurons that were responding to different kinds of stimulations, right? So that probably when we are beginning to learn how to write, how to read, there are different kinds of things that are taught to us, right? First, before we are taught alphabets, we are taught how to make standing lines, make sleeping lines, make slanting lines, right? Then make a curve, how to do this, how to do that. That is because we have to train different kinds of neurons for each of this, right? So once we have the hang of it, we can build them together and create character out of them, right? In addition, they found that neurons responding to signals from both eyes, but usually one or the other would dominate, right? So that is what. So you have two eyes, right? But whenever you are looking at something, you are not getting a double vision, right? Until and unless, there are some instances wherein, for instance, a person is going to faint, or a person is uh, facing some trouble, getting some kind of headache, migraine, that the person feels that they have double vision. Maybe because, both of the visual modalities, they are equally going into the brain, and the brain is not able to decide which one to, favor. Right. But generally, whenever you are looking at something, you are seeing a single vision, right? Because although you are looking from both eyes, one of the vision is getting dominated, and that is chosen in the brain itself, right? So, over subsequent years, Hubble and Wiesel refined their understanding, mapping the visual centers of their feline and primate subjects with increasing precision. Right? They found that visual cortex consisted of narrow columns of cells organized by eye preference and response to orientation, and they termed them as ocular dominance columns or orientation columns. Right? They combined, they formed an elegantly organized functional map of neurons that could process the complex input arriving from both of the animal's eyes, right? So this was one example, right? V1 is one of the centers. So these are four centers around cells, right? One, two, three, four. They are the centers around cells that are present. And a simple cell in V1, which is one of the regions of the brain, responds to lines of particular length and orientation, right? Which is this one. Its response may be derived from a combination of responses from different cells, which the centers around properties, right? These different centers around properties, they are going to the brain, and such as those located in lateral geniculate nuclei, and these kinds of cells are located in the LGN. Then there are these simple cells in visions, the cells that respond to light in a particular orientation, right? So it could be like photoreceptors themselves. Complex cells in vision, the cells that respond to light in a particular orientation but do not respond to single points of light, right? So that is why they are complex, because single point of light they will not respond to, but in a particular orientation, if it is coming, it will respond to it. Right. Then there are hypercomplex cells also, which will respond to particular orientations and particular lengths. Right. So these are further examples of convergent connections, which allow cortical neurons to abstract complex information from simple patterns also, right? For instance, this kind of a cell is giving an input that this is this kind of a line, while this, this way slanting is going, the information is going from this kind of a cell, it is going to the same neuron. So it is saying that it looks like this to me, right? Further, when it is sending that information to something else, that neuron is collecting information from multiple other neurons also. And based on the response of all these, it is building up whatever we are seeing to be a triangle, right? An upward triangle, a downward triangle, etc. Right? So that is what is happening over here. And that is how vision actually works. Now we come to sound, right? Which is another kind of sense. So this sound, it originates from the motion or vibration of an object, right? So we have, again, back in physics, we have already, studied the tuning fork, how if you move the tuning fork, it creates waves in the air, due to which you hear a certain kind of sound or a certain pitch of sound, right? For example, the vibration of vocal cords also within your larynx, and the plucking of a violin string, or passing of overhead aircraft. All of these are creating, waves of motion or vibration, due to which you are hearing something, or which is a sound to your ears, your brain, right? The human auditory system is capable of detecting a huge range of changes in the air, right? This pressure could range from around 0.00002 to more than 100 pascals, right? However, the role of the hearing brain is not merely to detect such changes, right? It also has to do with perceiving particularly what these changes mean, right? So as with vision, other perceptual systems, the goal of hearing is not to create a literal depiction of the outside world, but rather an internal model of the world that can be acted upon, right? So this model is constructed not only from ongoing sensory information, but also from previous sensory experiences, right? For instance, if you hear something and your eyes are closed, you are sitting in a closed room, you know that, okay, this somebody is ringing the bell of my house, right? But how do you know that? Because you have already stirred that this sound is coming from my house bell. Whenever the sound comes to me, it means that I have to answer the door. Similar for your phone ringing, right? Whenever this particular sound comes from my phone, I have to answer my phone, right? Because somebody is calling me. Or, for instance, you have not seen somebody, but you have just heard somebody say hello to you, right? And you instantly recognize that, oh, this is the friend that I was talking to earlier, right? So, all this is, constructed from previous sensory experiences also, right? There would be some time when you would have first heard that sound. It would not have meant anything to you, right? Somebody saying hello for the first time to you will not mean anything until and unless their voice resembles somebody. That is where previous sensory experience will get involved, right? So otherwise, the information has to be registered for these experiences to kick in, right? For example, we recognize a familiar tune when presented a different key, and we can recognize a familiar voice in a wide range of acoustic environments, right? So, for instance, the first thing that they are saying is, for instance, we know a song by heart, right? For instance, our national anthem, right? We know our national anthem by heart, right? I am not a musician, so I don't know which are the chords that it is played in. But for instance, I say that it has been played in, the G major, A minor, E major, something like that. These chords, right? So even if the chords are astray from the actual ones, instead of G major, E minor, and F major, if it is being played on E minor and some other chord altogether, right? But we know the tune is the same, right? We will still recognize it as a national anthem itself, right? So why? Because we are hearing a different sound altogether. But why are we able to put it together? Because of previous experience that our brain already has, and they are trying to figure out that okay, either it is a different instrument it is being played on, or a different kind of composition is being set to the same tune, something like that, right? So that is what is happening. Also, you're constantly outside, right? So even when you're, for instance, trying to talk on your phone, in a concert, right? It is hard. Why? Because there is music playing, there are people talking around you, other people's phones are ringing. There might be even the person you are talking to might have been in an environment wherein other noises are coming in, but your ear can actually filter, or rather your brain can actually filter what is the kind of thing you want to hear or focus your attention to, right? So in our noisy environments, our ears often encounter several sounds at once, right? But it is the job of the brain and not the ears to figure out how many different sound sources or streams there are and what they correspond to, right? So for instance, right now I'm speaking, I'm hearing my own voice, I'm hearing the fan, I'm hearing in the distance, the cars and the bikes passing by, their horns, everything I am hearing, right? But I am not actively being disturbed by them, right? Because my brain may not respond to that perception, right? If all this was not there, I might still be able to hear the, my watch go, the tick, right? My watch tick. But right now, I'm not being disturbed by it because my brain has other things to do, right? So this will depend both on incoming sensory information and learned knowledge about the sounds, right? Again, there's a difference between the sound that we hear and the auditory perception that we have, right? So, for instance, if one is listening to a familiar song, right? One of the familiar songs that I could think of was Waving Flag by Khan, right? So, there are a gap, but there is a gap of 2 to 5 seconds in the song, right? So, somebody might be, in the concerts, people do that, right? So, they have gone to a concert, the singer is singing their song, and they have sung 2 lines and then they have paused, right? Why have they paused? Because they want you to fill in, because it is a very familiar song, right? So if somebody in that tune sings when I get older, I, and then stops, right? The other person who is on the hearing end, if they are very familiar with the song, their brain will automatically fill in the gap, right? That older, I will be stronger, right? My brain will automatically fill that in. Okay, I may or may not have said it, but your brain will automatically fill it in if you are familiar with the song, right? So that is the brain that is coming into picture, right? The auditory perception that is coming into picture, that oh, this is that song, right? So then the auditory cortical areas are more active during the gaps relative to unfamiliar songs, right? Because if I'm hearing something for the first time, my brain is still trying to register, right? So these cortical areas where the experiences and memories already are, they will not be that active, right? Uh, our musical and lyrical knowledge can fill in silent gaps that are heard in songs, right? Sometimes that tune is coming, or you are hearing the musical version of a song, instrumental version it is known as, right? Instrumental version of that song, right? It is a song that you know by heart, right? You will still be able to, think about while you are listening to the instrument playing, you are still, your brain can fill in the lyrics by itself also, right? So another one is a difference that does exist between auditory and visual senses is their sensitivity to temporal and spatial information, right? So what temporal means with time and spatial means with space or with the distance that is being talked about, right? So the auditory system is an exquisitely tuned to detect temporal information. Right. So within time, as soon as information changes, you will be hearing it very quickly, such as rapid changes in frequency that characterize speech sounds, and in grouping information together over time, such as in extracting melody from music, right? The different time intervals associated with dots, dashes, and Morse codes are much easier to process when heard than when seen. Right, so Morse code, you can blink and you can do the Morse code, or you can tap or knock and do the Morse code also. So it is easier to grasp it when you are knocking it rather than when you are visually seeing it, right? In contrast, it is generally much easier to locate an object in space with vision than with hearing, right? So for instance, your eyes were closed and you are trying to, locate something by, in a room full of people where all the people are speaking and you have closed your eyes, you are trying to locate the person that you want to talk to, right? Versus if your ears were closed and your eyes were open and you were again trying to locate the person that you wanted to talk to, it would be much easier, right? To do that. Then comes the nature of sound, right? One of the simplest sounds has a sinusoid waveform. When the pressure change is plotted against time, it is going to be a sinusoid. And these sounds are termed as pure tones, right? So we'll quickly sift through this. Pure tones have a characteristic pitch that is related to frequency of sound wave. The human auditory system responds to sound frequencies between 20 hertz to 20,000 hertz, right? Below this will be infrasonic, more than this will be ultrasonic, right? Then the intensity of sound. That is its amplitude when considered as a sine wave is related to subjective experiences of loudness, right? For some people, the music that is playing in a room might not be that loud, but for others, it might be giving them a headache, right? So it is subjective again. We come to perception directly, right? So in perception, it is crucial to make a distinction between the physical properties of a stimulus and their perceived characteristics, right? Thus, in vision, there is a close relationship between wavelength of light, which is a physical property, and color, which is a psychological property. But the two things are not the same, right? So whenever we are seeing something as blue, right, it is because the kind of waves it is emitting to us are falling in that spectrum. It is not because that might be blue. It is a pseudo color actually, right? But it is possible to see color without associated wavelength, as in after images. And it is possible to process wavelength without perceiving color also, as in cerebral achromatopsia, right? So these are two different kinds of situations, after images and achromatopsia, right? Achroma, that means no color, right? So achromatopsia and after images are two examples of, disassociation of the color and the wavelength. But when we talk about sound perception, in hearing, although the pitch is related to frequency of sounds and loudness is related to intensity or amplitude of the sounds, right? When we actually hear, the pitch and loudness are regarded as psychological features of sound, whereas frequency and intensity are the physical properties, right? Although the amplitude and frequency might be independent physical properties of sound waves, the subjective properties most closely associated with them, that is pitch and loudness, are not processed by brain in a completely independent way, right? So the brain doesn't have two different processing centers for them. They process them pretty much together, right? In everyday life, pure tones are seldom heard, right? That is just a particular, particular pitch, particular amplitude. That is very less heard. However, many sounds can be described in terms of combinations of superimposed sinusoids of different frequencies, intensities, and phases. For example, musical notes typically contain a series of regularly spaced sinusoids. The lowest component is termed as fundamental frequency or F0 and typically determines perceived pitch of a musical note, right? The relative intensity of levels of different sinusoid components of the musical sounds are important for discriminating between the same notes played on different instruments, that is timbre, right? Then from the ear to brain, how is this passed, right? So we have been saying that the sensation is happening in the inner ear, the cochlea of the inner ear, right? This cochlea has hair cells which are perceiving this, sensing the sound waves, and the auditory nerve is attached to it, which is taking it to the brain, right? So the ear contains three main parts: the outer ear, middle ear, and inner ear, right? The outer ear contains the pinna, the earlobes, and the auditory canal. Reflections of sound waves within folds of pinna and within the auditory canal can amplify certain sounds and are important for locating a sound source. The middle ear converts airborne vibrations to liquid-borne vibrations with minimal loss of energy, right? And a series of three tiny bones are present in the middle ear, which are the malleus incus stapes or the hammer anvil stirrup, uh, which transfer mechanical pressure, which is, placed on the eardrum over here to basically at the end of the airborne auditory canal to smaller membrane called the oval window, right? It takes this pressure from here to oval window in the fluid-filled cochlea, right, which is transmitting it into this cochlea, which has hair cells. The inner ear contains chambers that are important for both the senses of hearing, that is cochlea and balance, which are the three semicircular canals. The cochlea converts liquid-borne sound into neural impulses. A membrane within the cochlea, termed as a basilar membrane, contains tiny hair cells linked to the receptors. Sound induces mechanical movement of the basilar membrane and hair cells on it. These movements induce a flow of ions through stretch sensitive ion channels that initiate neural activity and release of neurotransmitters. The basilar membrane is not uniform but has different mechanical properties at either end, and the nearest oval window is narrower and stiffer and shows maximal deflection to high frequency sounds. The end nearest the center of its spiral shape is wider and more elastic and shows a maximal deflection to low frequency sounds. And as such, different parts of the membrane are sensitive to different frequencies of the sound. Also, we can note over here that sounds originating from different parts of space do not stimulate different parts of the membrane, right? All of it is occurring in the same place. It is not like different photoreceptors are getting stimulated. The location of sound sources needs to be inferred from other kinds of information, such as differences between signals in the ears, etc. Right? For instance, if I'm hearing more intense signals from my right ear, the person must be on my right? He or she must not be on my left. So that is a kind of spatial derivation that we are getting out of it. Now, how is it going into the brain? This auditory nerve is going to the dorsal cochlear nucleus, which is, eventually going to the auditory cortex inside the brain. The ascending auditory pathway is not a passive transmission of information from the ear, but rather is involved in active extraction and synthesis of information in the auditory signal, right? So processing of the signal will happen in these regions that have been mentioned over here. There are 4 or 5 synapses in auditory pathway from the ear to brain, right? So this is one synapse, second synapse, third synapse, fourth synapse, right? Uh, yeah, at least what is depicted over here, right? Uh, So these will start from the auditory nerve to the cochlear nuclei in the brainstem and end with the projections from the medial geniculate nucleus to the primary auditory cortex, which is also called as A1 or the core region, right? The primary auditory cortex is located in Heshlus gyrus in the temporal lobes and is surrounded by adjacent secondary auditory cortical areas, which are called the belt and parabelt regions, right? These secondary regions receive some input from the medial geniculate nucleus and hence damage to the primary auditory cortex does not produce complete deafness, but does lead to problems in identifying location and locating the sound, right? So this is A1 and this is A2, right? The A1, which is primary auditory cortex, has these different kinds of frequencies which are in different regions of this cortex, and the secondary auditory cortex has the belt and parabelt areas, those, right? As we talked about. Yeah. So, while the cochlear nucleus has around 90,000 neurons, the medial geniculate nucleus over here has around 5 lakh, and auditory cortex has around more than 100,000 neurons, right? In addition, there are descending top-down pathways that can go as far back as cochlea itself, right? And may be important in auditory attention. The early auditory system can be said to have a tonotopic organization. That means, like it was mentioned over here, right? Different frequencies in different regions it is arranged, right? Different regions will have different uh, wavelengths, right? Just as different parts of the basilar membrane respond maximally to different sound frequencies, neurons within the auditory nerve respond maximally to certain sound frequencies more than the others, right? For instance, what is shown over here. The nerve bundle is orderly such that neurons responding to higher frequency are located on periphery and those responding to lower frequencies are more centrally located. To some extent, this organization is carried upward to early cortical stages also. So in the basilar itself, it has happened, but further, when we go to the cortex, again, it is happening over here. In both humans and animals, there is evidence that central region of primary auditory cortex responds to the lower frequencies over here. And outer regions on both sides to higher frequency, right? So that is how it is built. So when we compare the auditory and the visual systems, they have different routes of transfer, right? The thalamocortical route in auditory goes via the medial geniculate nuclei, which projects to the primary auditory cortex. While in case of the visual system, it goes via lateral geniculate nuclei and projects to the primary visual cortex. Then there is tonotopic organization in case of the auditory system, whereas there is a retinotopic organization, which is orderly mapping between position of retina and position of cortex in the visual system. Then the auditory system has more temporal sensitivity than spatial sensitivity, while visual system has more spatial sensitivity than temporal sensitivity. The auditory system is less well documented in the auditory domain, right? While visual system is well documented for color and movement. And there is evidence for separate auditory pathways for what versus where and how. That is where something is and how something is in the case of auditory perception versus what this is separate in separate pathways it is processed. And similarly, there is also evidence for separate visual pathways for what versus where or how in case of the visual system also. Then we come to touch, which is a third sense we are going to talk about, right? So touch is defined as the direct contact between two physical bodies, right? Something that we can, perceive with touch is something that is going to be tactile or it is going to be perceivable, right? It is going to be there in space, right? Something non-tangible we cannot touch. In neuroscience, touch refers to special sense by which contact with the body is perceived consciously, right? Touch can be active as when you move your hand or some other part of the body against another surface, or passive when someone or something else touches you, right? The passive touch is used for naming objects or describing sensations. Active touch is when used when hand manipulates objects, right? So, for instance, I'm moving this pointer by using my trackpad or my mouse, right? So that is active touch. But for instance, I am resting my hand on the arm of the chair. That is passive touch, right? I didn't mean to do it, but it is happening. So I know that this object is also present over here. Now I'm thinking about it. So now it might be classified as active also, but I was anyway doing it, right? Active and passive modes of tactile stimulation excite the same population of receptors in the skin and evoke the similar responses in afferent fibers. They differ somewhat in cognitive features that reflect attention and behavioral goals during the period of stimulation, right? So the hand has four types of mechanoreceptors. These four types are the Meissner corpuscles, Merkel cells, Pacinian corpuscles, and Ruffini endings. The sense of touch can be understood as a combined result of information provided by these four systems which are acting in concert. Right? So these are the Meissner corpuscles or RA1, Merkel disc receptor as SA1, Pacinian corpuscle or RA2, and the Ruffini ending, which is SA2, right? So each receptor responds in a distinctive manner depending on its morphology, innervation pattern, and depth in the skin. Receptors are innervated by either slowly adapting or rapidly adapting axons, right? So slowly adapting ones are SA, rapidly adapting ones are RA, right? So slowly adapting or SA fibers respond to steady skin indentation with a sustained discharge, right? For instance, I am, controlling this mouse. I have been doing it for over the past hour, right? So that is going to be slow adapting receptors that are, responsible over here. Rapidly adapting fibers stop firing as soon as indentation is stationary, right? For instance, um, I sat on the chair, right? Now, at that time, I felt that the chair was there, but now I'm not feeling that the chair is there, right? It has stopped continuously firing that, oh, this is chair, this is the chair, this is the chair, like that. It is not doing anymore, right? Sustained mechanical sensations from the hand must accordingly arise from the SA fibers, and the sensation of motion across the skin is signaled by the RA fibers. I hope the name itself is very clear, right? Then type 1 fibers terminate in clusters of small receptors in superficial layers over here itself of the skin at the margin between dermis and epidermis. And type 2 fibers innervate the skin sparsely and terminate in single large receptors in deeper layers of the dermis or in subcutaneous tissue. The RA1 fibers are the most numerous. RA1 was the Meissner corpuscles, which are present just below the skin, right? They are the most numerous tactile afferents in the hand, reaching a density of approximately 2 per millimeter square at the fingertip in man and monkey. right? These are the different kinds of receptors, the Merkel, Meissner, Ruffini, and Pacinian. There are the different kinds of locations that we have already visually seen. The different kinds of axon diameters, they have their conduction velocity. So the highest velocity is actually going to be all these three. This has a slower velocity and lower range of velocity also. And what is the best stimulus to activate it? For SA1, it is edges or points. For RA1, it is lateral motion. For SA2, it will be the stretch of the skin. For RA2, it will be the different kinds of vibrations that the skin might feel. Right? Then there might be different kinds of responses which can help to sustain the indentation, right? For SA1, because it is a slow adapting one, it is sustained with slow adaptation. For SA2 again, because it is a slow adapting fiber, it will be sustained. But for RA1 and RA2, that is not the case. It is not going to happen, right? Then there will be different frequency ranges also, which will be perceived by these different kinds of receptors. For instance, in SA1, it is going to be ranging from 0 to 100. From RA1, RA2, it will range for 1 to 300 or 5 to 1000, which are very, very high frequencies, right? Because over here we are, passing by that tactile information, or that much only we have to feed, right? So sustained information will anyway be slow. And the threshold for rapid indentation of frequency vibration will be best at around for around 8 micron and 40 micron for the slow adapting fibers, and it will be much lesser for the rapidly adapting fibers, right? So these are the receptive fields in the human hands, which are smallest at the fingertips, which means if the receptive field is smaller, meaning there will be more neurons over there. If the receptive field is larger, meaning the lesser neurons might be there, right? So the slow adapting mechanoreceptors, these are the receptive fields. Rapidly adapting mechanoreceptors, these are the kinds of receptive fields, right? Then the tactile information or touch information is processed in the central touch system. The central touch system comprises a dorsal column tract of the spinal cord, relay nuclei in the brainstem and thalamus, and a hierarchy of regions in the cerebral cortex. The tactile information enters the cerebral cortex through the primary somatosensory cortex, or the SI, in the post-central gyrus of the parietal lobe. This is the somatosensory cortex, right? This is the posterior parietal region, and SI. Yeah, this is the SI region, right? Or S1. The neurons in the S1 are at least 3 synapses beyond the receptors in the skin, right? For instance, in auditory, we saw they were at least 4 or 5 synapses beyond, right? In this visual, it was around 2 or 3 over there also. Their inputs represent information which has already been processed in the dorsal column nuclei and the thalamus and the cortex itself. Sorry. Each cortical neuron receives inputs arising from receptors in a specific area of the skin, and these inputs together are its receptive fields, right? We perceive that a particular location on the skin is touched because a specific population of neurons in the brain is activated in response to that touch. This experience can be experimentally induced by electrical stimulation of the same cortical neurons, right? The primary somatosensory cortex comprises 4 cytoarchitectural areas, which are the Brodmann's areas, termed as 3A, 3B, 1, and 2. So these are the 4, 3A, 3B, 1, and 2, right? In the S1 region. And then there's a S2. These areas are extensively interconnected such that processing of sensory information at this level involves both serial and parallel processing. The neurons in 3 and 3B receive input from the lateral and medial zones of the ventral posterior nucleus of the thalamus and project axons into areas 1 and 2, which is represented over here. The areas 3B and 1 receive information from receptors in skin, whereas 3 and 2 receive proprioceptive information from receptors within the body, that is muscles, joints, skin, etc. Somatosensory information is conveyed in parallel to the four regions of S1 to higher centers in cortex, including the secondary somatosensory cortex, which is the S2, the posterior parietal cortex, and the primary motor cortex, right? And there is columnar organization of the somatosensory cortex, right? You can see that there are multiple columns over here that they have to pass, right? So that is what this organization entails. Yeah. Though the sense of smell and taste, through the sense of smell and taste, we are basically okay. So that was the end of the touch one, right? And these two are the senses that were left, the smell and the taste. And these two are pretty much interlinked, right? For instance, when we will talk about flavor, it will not just be the taste which is giving you the flavor or the smell that is giving you the flavor. It will be a combination of the two, right? So it is very, very interlinked, these two senses. Yeah, did somebody say something? So, when we talk about the sense of smell and taste, we are able to perceive a staggering number and variety of chemicals in the external world through these two senses that we have, right? The chemical senses inform us about the availability of foods and the potential.
আমরা জিদা রান্না করে আগুন জ্বালিয়ে দিয়েছে
Just wait. I'll share again. Yes. Yeah. So these senses together, they will inform us about whether there is potential danger or potential pleasure in consuming those foods, right? For instance, there are some, that will have a very, very chemical, volatile, pungent kind of smell, and just by the smell, you will not feel that it is appetizing, right? So it will tell you that probably it will not taste that well, or probably it is not going to be good for your body, something, right? The smell and taste also initiate physiological changes required for digestion and utilization of the food. In many animals, the olfactory system also serves as important social function by detecting pheromones that elicit innate behavioral or physiological responses. For example, in the honeybees or in the insects, right? There are a lot of pheromone-based responses that are happening, and that is through the olfactory system, right? Also, sometimes we start masticating, or our mouth starts watering, our salivary glands get activated as soon as we smell food. We have not even had food, but as soon as we smell food, uh, it gets activated, right? So that is also going to aid in, digestion of through the releases of amylases when we are going to consume the food. Then large number of olfactory receptor proteins initiate the sense of smell also. Odorants are volatile chemicals that are perceived as odors and are detected by olfactory sensory neurons in the nose. These sensory neurons are embedded in specialized olfactory epithelium over here that lines part of the nasal cavity, approximately 5 centimeter cube in the area in humans, and is interspersed with glia-like supporting cells, right? And it is very closely linked or in very close connection to the brain itself also, right? And these are the supporting cells that were being talked about, right? Glia-like supporting cells in this olfactory bulb and epithelium. The olfactory sensory neuron is a bipolar nerve cell. A single dendrite extends from the apical end to the epithelial surface, where it gives rise to numerous thin cilia, right? So these are the cilia. This is the single cell that is extending from this surface to that surface. The cilia protrude into the mucus that coats the nasal cavity, right? So these are the cells that are present here, like cells that are present in the nasal cavity. The cilia have receptors that recognize odorants as well as transduction mechanism needed to amplify the sensory signal and transform them into electrical signals in the neuron's axon, which protects from the basal pole of the neuron to the brain, right? So, for instance, this was a nasal epithelium. This is the cribriform plate, which is a tiny, separation between the nasal epithelium and the brain, olfactory bulb that is in the brain, right? And these are the regions of the brain itself. Axons of olfactory sensory neuron pass through the cribriform plate, a perforated region in the skull above the nasal cavity. The axons then synapse in the olfactory bulb, and these are the olfactory bulb. Over here are different glomeruli in which they are going to synapse with tufted cells, mitral cells, etc. And these are going to pass into the olfactory pathway, right? The humans have approximately 350 different odorant receptors, whereas mice have approximately 1,000 odorant receptors, right? They are more dependent on sense of smell. We are a little less dependent on sense of smell. Although odorant receptors belong to the G protein coupled receptor superfamily, they share sequence motors not seen in other superfamily members, right? So, although they are GPCRs, but these odorant receptors are very specialized kind of GPCRs, right? Significantly, odorant receptors vary considerably in the amino acid sequences also. A second smaller family of chemosensory receptors is also expressed in the olfactory epithelium itself. These receptors are called the TARS or the trace amino acid receptors, right? They are also GPCRs, but their protein sequence is unrelated to that of these other odorant receptors that we were talking about that were very unique. They are encoded by a small family of genes present in humans and mice as well as fish, right? Now, these are different kinds of odorant receptors, right? So, depending on which combination of receptors get activated, it will give you different kinds of odors, right? For instance, when only this purple one is activated, it gives you a rancid, sour, boat-like odor. When along with purple, all these other receptors are also activated. It gives the rancid, sour, and sweaty kind of odor. But if with this red and green are activated, it will give you a violet, sweet, and woody kind of odor, which is almost totally opposite of when only this receptor was getting activated or only when these two were getting activated, right? So that is what. So the combination of odorant receptors that get activated will decide which is the particular kind of smell that we are able to sense, right? Odorants are encoded in the nose by dispersed neurons. Different types of odorant receptors are expressed in different coarse zones of olfactory epithelium of the mouth also. Right? Each receptor type is expressed in approximately 5,000 neurons that are confined to one zone. The neurons with the same receptors are randomly scattered within the zone so that the neurons with different receptors are interspersed. Although one zone may have more receptors for a particular odorant compared to other zones, all zones contain a variety of receptors so that a specific odorant may be recognized by receptors in several different zones. Right? Because each of these odorant is detected by an ensemble of neurons that is widely dispersed across the epithelial sheath, right? For instance, this particular odorant is present in these regions, this is present in these other regions, right? When part of the epithelium is damaged by respiratory infection also, a particular odorant will remain functional, right? Then axons of olfactory sensory neuron project to the ipsilateral olfactory bulb whose rostral end lies just above the olfactory epithelium. The sensory axons terminate on the dendrites of this olfactory bulb within bundles of neuropile called glomeruli that are arrayed over the bulb's surface. I think I have not kept the taste receptor within this presentation. Give me 2 minutes. I'll just add on. I thought maybe this is all I'll be able to cover. Yes, I'll continue. Yes. So we were here. These sensory inputs are arranged by the receptor type, right? Axons of olfactory sensory neurons project to ipsilateral olfactory bulb, and whose rostral end lies just above the olfactory epithelium. Sensory axons terminate on the dendrites of olfactory bulb neurons within the bundles of neuropil called the glomeruli that are arrayed over the bulb's space, right? One second. Yeah. In each of these glomerulus, the sensory axons make synaptic connections with the three different types of neurons, either the mitral and tufted relay neurons, which project axons into olfactory cortex, and the periglomerular interneurons, which encircle the glomerulus, like this, right? The axons of an olfactory sensory neuron terminates only in one glomerulus, and similarly, primary dendrite of each mitral and tufted relay is confined to a single glomerulus. Each glomerulus, the axons of several thousand sensory neurons converge on the dendrites of approximately 40 to 50 relay neurons. And this convergence results in approximately 100-fold decrease in number of neurons transmitting olfactory signals to them. So this is a representation of axons of different neurons with same odorant receptors converging on a few glomeruli, right? One kind of odorant is being supplied. So this is the glomerulus getting activated. Another is getting supplied. These are the glomeruli that are getting activated. While one odorant can activate many glomeruli also, for instance, what is seen over here, right? Odor responses in the olfactory bulb. The organization of sensory information in the olfactory bulb is dramatically different from that of the epithelium, right? How is it different? Because olfactory sensory neurons with the same odorant receptors are randomly scattered, right? Like this blue one is one kind of receptors, red one is another kind of receptor. So they are very randomly scattered in one epithelial zone. Their axons converge in a few glomeruli at two spots, one on either side of the olfactory bulb, right? So over here and another glomeruli on the other side of the olfactory bulb. Each glomerulus and each mitral and tufted relay neuron connected to it receive inputs from just one type of odorant receptor. I think that is clear by now. The result is a precise arrangement of inputs from the different odorant receptors, which is similar between individuals. And because each odorant is recognized by a unique combination of receptor types, each odorant also activates a particular combination of glomeruli in the olfactory bulb. At the same time, just as an odorant receptor recognizes multiple odorants, a single glomerulus or a given mitral valve or tufted cell is activated by more than one odorant. This organization of sensory information in the olfactory bulb is likely to be advantageous in two different respects. First is the fact that signals from thousands of sensory neurons with the same odorant receptor types always converge on the same flow glomeruli and relay neurons in the olfactory bulb may optimize the detection of odorants present at low concentration. And each glomeruli is encircled by periglomerular interneurons over here that receive excitatory input from sensory neurons and form inhibitory dendrodendritic synapses with the mitral and tufted cell dendrites in that glomerulus and perhaps adjacent glomeruli. The periglomerular interneurons may therefore have a role in signal modulation, right? They are modulating the signal by giving some kind of an inhibitory signal also. Granule cell interneurons deep in the bulb provide negative feedback onto mitral and tufted cells, right? Over here also, this is also giving negative feedback. Granules and interneurons are excited by the basal dendrites of mitral and tufted cells and in turn inhibit those postsynaptic relay neurons and others in which they are connected. The lateral inhibition afforded by these cells, these connections is thought to dampen signals from glomeruli and relay neurons that respond to odorant only weakly, right? So there will be some very weak smells also. They don't want those weak signals also to get through. They want us to be able to distinguish signals without getting confused. So that is why this kind of an inhibition is also happening, thereby sharpening the contrast between the important and irrelevant sensory information before its transmission to the cortex. Olfactory bulb transmits information to the olfactory cortex, right? So this is olfactory bulb through different regions. It is finally taking the information to the olfactory cortex, right? Then we come to taste. Taste has 5 submodalities of qualities. The primary function of gustatory system is nutritional itself. Humans and other mammals can distinguish 5 different taste qualities, which are sweet, bitter, salty, sour, and umami, the taste associated with amino acids. The taste chemicals or taste tints perceived as sweet are associated with food high in caloric content, while those sensed as umami are indicative of protein. Consistent with nutritional importance of carbohydrates and proteins, both sweet and umami taste elicit pleasurable sensations in humans and are attractants for animals. In contrast, the bitter taste tints, which are often found in poisonous plants, elicit an aversive response that is innate in human as well as animals as well as human infants and likely prevents the ingestion of toxic substances, right? So, especially when we are younger, we will not eat something that is bitter, right? Even the bitter gourd, karela, and all these sabzis we don't like as children, right? Because we have to build a taste for it. Because by default, our taste buds are not accustomed to that, and our wiring is such that we are built to not like it, right? Because it might be harmful, the bitter tasting things, right? So then we come to this discussion of taste versus flavor. So taste is often thought to be synonymous with flavor, but that is not the case. Taste refers strictly to 5 qualities encoded in the gustatory system, whereas flavor, with its rich and varied qualities, actually stems from a combination of inputs from both the gustatory, olfactory, and even other somatosensory systems. In the gustatory system, sensory signals generated in mouths are relayed through the brainstem and thalamus to the gustatory cortex. The tastings are detected by taste receptor cells that are clustered in the taste buds. Although the majority of taste buds in humans are located on tongue, some can also be found in the palate, that is the upper palate, the pharynx, the epiglottis, and the upper third of the esophagus also sometimes. Taste detection occurs in the taste buds mostly over here, and then it is relayed to this area in the brain, right? To the gustatory cortex. The taste buds are clustered in papillae on the tongue. Taste buds on the tongue occur in structures called papillae, of which there are 3 types based on morphology and location, right? So they could either be circumvallate like this, or foliate like this, or fungiform like this. Each fungiform papillae contain 1 to 5 taste buds, while circumvallate or foliate papillae contain hundreds of taste buds. The taste bud in garlic-shaped structure embedded in the is a garlic-shaped structure embedded in the epithelium, a small opening at the epithelial surface. The taste pore is the point of contact with the tastids, right? This is what it looks like in electron microscopy or in a great, histology image. I'm assuming histology it looks like. Yeah. The taste buds also contain other elongated cells that are thought to serve as supporting function, as well as small number of round cells at the base which are thought to serve as the stem cells. Taste cells are short-lived and appear to be continually replaced from the stem cell population. Although these taste cells are non-neural, their contacts with the gustatory system, sensory neurons, have the morphological characteristics of chemical synapses, including clustered presynaptic vesicles. The taste cells also resemble neurons in that they are electrically excitable. They have voltage-gated sodium, potassium, and calcium channels and are capable of generating action potentials as well. So each of these 5 taste qualities involves a different sensory transduction mechanism in the microvilli of taste cells. There are, however, 2 general types. Bitter, sweet, and umami tastings interact with GPCRs, whereas salty and sour tastings appear to involve in specific ion channels. So there are 2 different kinds of ways it can happen in, right? So again, bitter, salty, sour, and salty and sour, they will have the ion channel base, and bitter, sweet, and umami will have the GPCR base, right? And these are how different kinds of receptors combinations are, able to perceive different kinds of tastes. Each taste cell is innervated at the base by peripheral branches of the axon of the primary sensory neurons. Each sensory fiber branches many times, innervating several taste cells within the numerous taste buds. The release of neurotransmitters from the taste cells onto the sensory fiber induces action potentials in the fibers and transmission of signals to the sensory cell bodies. These cell bodies of gustatory neurons lie in the geniculate petrosal and nodose ganglia over here. Petrosal, nodose, and geniculate ganglia. The peripheral branches of the gustatory sensory neurons in these 3 ganglia travel in the cranial nerves 7, 9, and 10, right? And eventually, the central branches of axons of gustatory sensory neurons enter the medulla, where they terminate on the neurons in the gustatory area of the neurons of the solitary tract. In most mammals, neurons in this nucleus transmit signals to the parabrachial nucleus of the pons, which in turn sends the, this is a gustatory area, it goes to the parabrachial area of the pons, which is going to send it to gustatory information into ventroposterior medial nucleus of the thalamus. And in primates, these neurons transmit gustatory information directly to taste area of the thalamus. From the thalamus, taste information is transmitted to gustatory cortex over here, located along border between the anterior insula and the frontal operculum. The gustatory cortex is believed to mediate the conscious perception and discrimination of taste stimuli. The taste area of the thalamus also transmits information both directly and indirectly to the hypothalamus, a structure that controls feeding behavior as well as autonomic responses. Yeah. So I know I have sped up with it. A lot of information I have just kept here for your understanding, right? You don't necessarily need to retain that information. It will be helpful for you to understand the logic behind it rather than retain that information, especially in this portion, right? But for the first 2, that is the visual and the sound perception, that is where I had spent most of the time in building the concepts, and more similar, like the concepts remain more or less similar as we go across all the others also, right? So, yeah. That is the end from my side for the course also. Yeah. So in case you have a problem or have a question, query, you can go ahead and ask. Okay, so if not, I'll just stop the recording and we'll end the session. Thank you so much for joining. It was a final session for this course. And if you have registered for your examinations, all the very best to you for that. And I hope it goes well. Thanks. All the best.
Thank you,
Okay.
ma'am.